In this section, we're going to discuss catalysts and enzymes. Catalysts work to speed up chemical reactions. They do this by lowering the activation energy of the reaction. As a result, less energy is needed to start the reaction, so it happens more quickly. Catalysts remain unchanged in the reaction. The initial and final energy of the reactants and products is also not changed by the catalyst. So what do catalysts do, or how do catalysts work? They work by decreasing the activation energy. So if we look at the image of this rock being pushed over the hill, we can't just quickly push the rock up and over the hill. If we think of that hill as the activation energy, a reaction cannot quickly occur unless the molecules possess sufficient energy, known as the activation energy. If the height of the hill were lower, like you see here, the person could roll the stone over the hill at a faster rate. Catalysts work in much the same way. They speed up reactions by reducing the activation energy for the reaction. Energy of life. Life requires energy. Living things take in energy, transform it, and release it. This is accomplished by chemical reactions. Chemical reactions in living things need to be very fast. Enzymes are biological catalysts, or catalysts made by living things. They are almost always proteins. Some RNA molecules, called ribozymes, act as enzymes, a fact discovered in 1982 by Sidney Altman and Thomas Cech. Currently, RNA catalyzes over 100 cell reactions. All cellular life uses enzymes to speed up chemical reactions. Enzymes lower activation energies and speed up chemical reactions in living things. How enzymes work. Enzymes increase the speed of a reaction by decreasing the activation energy of the reaction. The energy of the reactants and the products is the same. So if we look at this graph, we have energy on the y-axis. We have the energy for the reactants and the energy for the products. That is unchanged, but you can see by the red dotted line with the catalyst, the hill or the activation energy is lowered. Since the energy of the reactants and products is the same, the reaction's Gibbs free energy, or delta G, this energy between the reactants and the products, is also unchanged. Naming enzymes. Many enzymes' names end with the suffix ase. Examples are lactase, amylase, and peptidase. We will learn about these common digestive enzymes on the next few slides. Common enzymes. Lactase is an enzyme that helps us to digest dairy products. The sugar found in dairy products is a disaccharide, lactose, which you can see here. The enzyme lactase breaks down lactose into two monosaccharides, galactose and glucose. People with lactose intolerance have trouble digesting dairy products because they stopped producing lactase in adulthood. People who don't make lactase can take lactate pills that contain the enzyme lactase before eating dairy products. Amylase is an enzyme found in human saliva. It begins the process of carbohydrate digestion in your mouth. As you chew, amylase breaks down starch into a disaccharide, maltose. Potatoes are starchy. Starch is made of many sugar molecules which are bound together. When you chew a potato, amylase breaks down the starch into smaller sugar molecules called maltose. It's part of digestion. Your body makes amylase. So you can see we have this long starch from maybe a potato, and this amylase molecule breaks the starch up into smaller disaccharides known as maltose. It would take years to break down this starch without the help of this amylase enzyme. Peptidases are a family of enzymes that break down proteins into polypeptides and amino acids. Peptidases catalyze the hydrolysis reaction that breaks peptide bonds. Peptidase 1, an allergen found in dust mite feces that causes human allergies, is pictured here. So, how do enzymes decrease activation energy? Enzymes speed up biochemical reactions by providing a space called an active site for the substrates or reactants to bond. This decreases the activation energy required for the reaction by putting the substrates in the right position to have their bonds broken and then remade again to form the products. Keeping the substrates close together enables them to react quicker than if they were just randomly drifting through the environment. Furthermore, reactions require the formation of a transition state, which is an intermediate molecule between the reactants and the products. Transition states have a higher energy than either products or reactants because they are unstable. The active site provides a chemical environment that helps stabilize the transition state, thus lowering its energy and speeding up the reaction. Just as a key only opens a specific lock, each enzyme has its own unique shape, so each enzyme is specific to certain substrates. As the substrates enter the active site, labeled A1, so this is the active site of the enzyme, the enzyme's shape changes just a little bit. So as these two substrates, B1 and B2, enter the enzyme's active site, the enzyme's shape changes just a little. As you can see here, it's kind of curved around in order to create a better fit for the two substrates or reactants, remember, B1 and B2. And this is known as an induced fit. Enzyme substrate complex. When the substrates fit into the enzyme, it is called an enzyme substrate complex. The reaction takes place and the products leave the enzyme. So here's the active site of our enzyme, and here's the substrate entering the active site. Then we have our enzyme substrate complex. This is where the reaction is able to take place between our substrates. So our substrates or our reactants form products. And then we have our enzyme product complex. Finally, the products are able to leave the active site of the enzyme. Here's another view of the enzyme function. Here in step 1, the active site is available for a molecule of substrate, the reactant on which the enzyme acts. This is the enzyme sucrase. The substrate then, sucrose, then binds to the enzyme here. With the addition of water needed for the hydrolysis reaction in this case, the substrate is converted into its products, and then those products are released as fructose and glucose. And then our enzyme is available again to repeat the cycle. Amino acid impact. The types of amino acids that make up enzymes are key to their shape and functionality. The amino acids interact with the environment and with one another. The unique amino acid side chain, or R groups, determines the chemical properties of the amino acid. They may be nonpolar, positively charged, negatively charged. The electric charge on the enzyme's amino acids at the active site must be opposite to the charge of the substrate. Opposite electrically charged substances attract each other, while substances with the same charge repel each other. Enzyme shape and functionality. The shapes of biomolecules rely on interaction between chemical groups. In enzymes, the chemical groups in the backbone and the chemical groups in the side chains give rise to secondary, tertiary, and quaternary structure. In an enzymatic reaction, like the ones we've been discussing in this topic, interactions between chemical groups of the enzyme and substrate change their shapes. As they interact, bonding patterns in the substrate will be permanently changed, leading to the formation of products that are different from the substrate. To help you out, here's a review of the levels of protein structure. The different structures of the enzyme are determined by the amino acids. Primary structure. Each enzyme consists of a unique amino acid sequence held together by peptide bonds. The order is important because it determines how they fold. Secondary structure. The hydrogen bonds between amine groups, the nitrogen-hydrogen groups, and carbonyl groups, the carbon double bond oxygen groups, in the backbone are what maintain these shapes. R groups do not play a role in the shape. Tertiary structure. This is driven by the interactions of R groups with one another and their environments. R groups cluster or repel due to hydrophobic and hydrophilic interactions. They attract or repel each other due to charge interactions. Quaternary structure. This consists of multiple polypeptides bound together, frequently with disulfide bridges. Not all enzymes have this structure. Quaternary structure is key to enzyme regulation and activity, to be discussed later. For example, take an enzyme that is in charge of cleaving the phosphate group off cyclic AMP. So here's the primary structure of this enzyme, just the chain, unfolded chain of amino acids. This enzyme folds into its quaternary conformation, or the folded protein that you see here. The protein folds in such a way that 6 highlighted R groups from these different colored circles end up in the binding site of the enzyme. This is important because if the protein folded any other way, the active site would be chemically different due to the presence of different R groups. For example, they could create a nonpolar environment instead of a polar one in the active site. Let's now zoom in into the enzyme's active site. Remember those colored R groups from the previous slide? You can see them in more detail here. So here's 1, 2, 3, 4, 5, 6. Here's our cyclic AMP, our substrate. This particular enzyme's active site contains several polar R groups that hydrogen bond, you can see one here, with cyclic AMP and a positively charged R group, here's the one you see in yellow, that interacts ionically with the negatively charged phosphate group, so here's our phosphate group, in the substrate. The hydrogen bonds and the electrostatic interactions generated by the R groups hold the substrate in place so that the enzyme can carry out its catabolic function. Furthermore, the interactions between the active site and cyclic AMP have a twofold effect. The active site changes slightly to better fit cyclic AMP, that's the induced fit that we talked about before, and cyclic AMP is bent in a way that makes the cleavage of the phosphate group easier. The bent form of cyclic AMP is its transition state.